All right, welcome. So it's my pleasure to kick off Grand Rounds this morning, and as always, I want to draw your attention to the wonderful program that we have coming up. There's some real high points here. So our Richard Berman lecture, it's a big honor, this particular lectureship, so re-envisioning medical education, challenging the status quo. I think Becky will be down here in the front row for that one, right? And then this looks incredible, a decade of open notes, how transparency and participation can transform healthcare. I'm actually especially interested in that because it was about a decade ago when my husband, who led this at Penn for releasing radiology reports, I remember how incredibly controversial it was. So it'll be very interesting to see that. And then finally, the resident house-wide debate, which is always a great event. So mark your calendars. And then we told you last week about this upcoming perinatal potpourri event that down in Monterey with an all-star cast of faculty that you can see here. And then again, as always, a really exciting agenda on the ENGAGE program. So healthy work environments through crucial conversations. That's actually quite interesting because I went back and read the whole task force report on department work environment and decision-making and transparency, and going to be talking about that at the faculty meeting on Monday. And the whole idea of a healthy work environment is a big theme in that. So I'm looking forward to learning more about that. And then Sarah's going to be talking about curriculum development and measuring outcomes, part of this series. Pediatric research retreat is around the corner. This is a really wonderful event. I strongly encourage you to come, not for two reasons. One is to hear about some of the phenomenal work that's going on among our faculty. It's a chance to hear from some of our new faculty like Sahel, and and hear more. And then for the faculty who've been here for some time like Lee, I think often the division chiefs and some of the senior faculty are doing incredible work. People aren't always aware about the amazing things that are going on in the department. And then some of our rising stars, our junior faculty like Katie Travis, Robbie Majner, and then you can see a really great representation of our entire mission. But another really important reason to come to this is it's really the highlight of the year for our trainees, for our postdocs, and for our fellows. So strongly encourage you to come to the research retreat in April. And with that, we'll, as always, put up the planner and faculty disclosure to learners, and then Megan's going to introduce our distinguished speaker.
Bring this down for myself. I'm very honored to introduce our speaker today, Dr. William Poo. He's here for our pediatric cardiology visiting professor program. Dr. Poo is a practicing cardiologist and the Aldo Castaneda Professor in Pediatrics at Harvard Medical School. He's also the Director of Basic and Translational Cardiovascular Research in the Department of Cardiology at Boston Children's Hospital. Dr. Poo's lab focuses on regulation of gene expression and signaling in cardiac development, disease progression, and regeneration. And he's going to be telling us today about some things he's learned from his team's modeling of two forms of inherited heart disease. So please welcome Dr. Poo.
Hey, thank you for that introduction, and for inviting me to give this talk and having a chance to share some of the work that we're doing. The disclosures. So over the last 50 years, pediatric cardiology has made really remarkable advances in the treatment of structural heart disease. So this is a graph of the mortality in congenital heart disease. This is actually data from Norway. From 1970 to 2010, and you can see the amazing reduction that we've been able to achieve in the mortality rate. However, we haven't done so well with muscle disease. So really there has not been much advance in improving the function of heart muscle other than transplantation and mechanical support. So one of the reasons for this is that it's difficult to study human heart tissue. So as you can imagine, human heart samples are not obtained frequently, especially in pediatrics. We can't maintain heart cells in culture. And we often use mice to model heart disease, but mice are not perfect models of human physiology. For example, they're 2,000 times smaller. Their stroke volume is about 2,000 times less, and their heart rate is almost 10 times higher. Duplicate. So it was a huge advance when in induced pluripotent stem cells technology was developed. This is the original paper from Yamanaka that described how you could take somatic cells and put a magical cocktail of 4 factors onto them and turn them into stem cells. And then these stem cells could be renewed indefinitely or directed to differentiate into many other cell types, including cardiomyocytes. So this achievement has really transformed how we do research, and as a result, within 6 years, it was awarded a Nobel Prize. So just as an example of how this has changed what we do, this has really become like one of the bedrock technologies of the lab, so that you can actually buy kits off the shelf that will turn somatic cells into iPS cells. You can also buy kits that will direct the differentiation of iPS cells into cardiomyocytes. So why have iPS cells been so transformative? So this slide shows some of the uses, both current uses and future imagined uses of these cells. So you can take cells from a patient and make iPS cells from them, turn them into different types of cells like cardiomyocytes, and they can be used for disease modeling to discover new disease mechanisms, or they can be used to screen drugs to develop new therapies or discover toxicities in existing therapies. And an important feature of these iPS cells is that they share the genetic material of the person who donated the cell. So here, as an example, you have people with 2 different genotypes, and the iPS cells derived from them will share those same genotypes. So then if you were to use this to guide a clinical trial, you could imagine first doing the clinical trial on the iPS cells to select the patients that might be most responsive, and in that way de-risk the entire trial. In clinical medicine, so this is not something that we've achieved yet, but it's something that's hoped for in the future. It's thought that maybe these cells would provide personalized disease models that would, so right now we treat each patient sort of like an independent experiment, N of 1, and you have to do trial and error to find the right treatment. But with the iPS cells from the patients, maybe you could design or select the right therapy for the patient in a dish and then apply the therapy to the patient. But there are a lot of barriers to using iPS cells at this stage for studies in the heart as well as in other organ systems. One of the most important barriers is these cells are very immature. They resemble fetal or neonatal cells rather than adult cells. So this picture shows what an iPS-derived myocyte looks like and what the real thing looks like. You can see they're quite different. We have become pretty good at turning iPS cells into these immature myocytes, but you know, the heart is not just myocytes, an organized array of many different cell types assembled into a tissue. We're not yet very good at making those tissues. We also don't have good physiological assays of tissue and organ function. And finally, I think a big question mark in the field remains central to, you know, the hope for these cells is that they will actually model the responses of individual patients. That's something we can hope for, and theoretically it may occur because of the shared genotype of the cells in the patients. But the extent to which that's true is, I think, yet to be established. So in this talk, I'll just share with you our work in making engineered human tissue models of 2 different types of heart disease. One is a form of cardiomyopathy that's called Barth syndrome, and the second is an inherited arrhythmia known as CPVT. So generally, when we use iPS cells, we take the cells from the patient and we engineer the cells into a disease model. We then try to understand the mechanism by which the patient's mutation causes their disease. And from understanding the mechanism, we tried to develop a candidate therapy, which we can then bring back to the patient. So in the first example, we studied a disease called Barth syndrome, and all of this work was a close collaboration with a bioengineering group led by Kip Parker. So this started when I was a cardiology fellow, and sorry, the date discloses my age, but I saw a 2-year-old that was admitted to the medical service with respiratory distress, and the patient was diagnosed ultimately with a severe dilated cardiomyopathy. And a standard part of that workup is to do urine organic acids, which reveal 3-methylglutaconic aciduria. So this is a characteristic finding of this disease, Barth syndrome, which is an X-linked mitochondrial myopathy. It affects both skeletal and heart muscle. And the gene that's mutated is pathogenesis. So this is a rare disease. It occurs about 1 in 300,000 live births, but it does affect about 3 to 5% of boys that have cardiomyopathy. And about 5 to 10 new cases are diagnosed each year. So tafazin is an enzyme that catalyzes the maturation of a lipid that's called cardiolipin. This is the signature phospholipid of the inner mitochondrial membrane. So here's the reaction that it catalyzes. You can see it adds an acyl chain to this MLCL to give you the tetra acyl form. So this form of cardiolipin is required for the normal function of all the enzymes that are in the inner mitochondrial membrane. So here's our approach. We got the skin sample from the patient. At that time, we were doing skin biopsies. Now we're using peripheral blood. We made iPS cells. We turned them into heart muscle and into cells, and you can see that we can get these sheets of beating cardiomyocytes. So I'll just summarize the work we did to characterize the phenotype of these, the metabolic phenotype of these cells. As you would expect, the cells had abnormal cardiolipin profile, abnormal oxygen consumption, and other markers of abnormal mitochondrial function. We were able to correct these metabolic defects by replacing the mutant gene, which shows that this gene mutation in the cells is what's causing the problem. And we were also able to use genome editing to introduce the mutation into wild-type cells to show that the mutation itself was sufficient to cause the disease rather than having to work with modifier genes. So next, we wanted to look into the reason that this mutation would cause a contractile problem. So here again is iPS cells that are derived from a wild type, but you can see that the sarcomeres are poorly organized, which makes it difficult to study the assembly of sarcomeres in patient cells. So instead, we used contact printing to help the cells to get more organized. So we basically stamped rectangles of fibronectin, which the cells like to sit on. And then as a result, you can get the cells to assemble much more organized sarcomeres. And what this image shows is that the wild type cells could make quite organized sarcomeres, but in these Barth cells, they had a lot of trouble making these sarcomeres. And if we put the gene back, then we can restore sarcomere organization. But the key cardiac phenotype of these patients is a cardiomyopathy. So we wanted to be able to measure the function of the cardiomyocytes, and not to do it in single cells, but to do it in an engineered tissue, because the heart is not just individual cells, but they're organized into a tissue. So we put the cells on a flexible plastic sheet that was lined with fibronectin, which made the cells have an orientation. So with this orientation, they develop organized force, and they'll actually bend the sheet when they contract. So since we know the stiffness of the sheet, we can measure the force that's being produced by the cells. So here the red line is a computer-generated tracker to just help you see the movement of these sheets. So with this heart-on-a-chip model, we compared the control cells to the Barth cells. You can see that these cells are having a lot of difficulty contracting. If we put the TAS gene back, we can restore the contraction. So that tells you that we didn't just somehow screw these cells up, but this is a real phenotype related to the lack of tafazin. So now that we have a model of this cardiomyopathy, we're able to ask the question, what is the link? What's the molecular link from this defect in cardiolipin metabolism with the contractile defect? And one important piece of data that I'm not going to show you is that when we correct the ATP deficit in these cells, we do not restore the contractile function, suggesting it's not a simple lack of energy. So if we dive into the mitochondria, in the inner mitochondrial membrane, there's elaborate machinery that converts fuel like acetyl-CoA and oxygen into energy. Cardiolipin is important to hold this machinery together so that in the absence of cardiolipin, the machine becomes inefficient and generates a large amount of reactive oxygen species. So we hypothesized that maybe this reactive oxygen species is what's linking the cardiolipin defect to the contractile defect. So we used a mitochondrially targeted reactive oxygen scavenger to test that hypothesis. So here we're measuring the amount of ROS production in these cells, and you can see that the mutant cells produce a large amount of ROS, which can be normalized by adding the mitotempo. So we added the mitotempo and did the sarcomere assembly assay, and you can see that it actually corrects the sarcomere assembly of these cells. We then did the heart-on-a-chip assay, and you can see that when we add mitotempo, we don't have any effect on the wild-type cells, but it restores the contractile function of the Barth cells just as well as adding tafazin did. So this actually led to a biotech company to do the first clinical trial in Barth syndrome of this compound elimipritide, which they claim is a ROS scavenger. And this trial recently completed. They haven't fully reported the data, but they have said that the secondary endpoint of cardiac stroke volume was improved by the treatment. We've been interested in TAS gene replacement therapy because of this experiment in which we showed that adding TAS back can correct the problem. So this leads us to AAV gene therapy. So adeno-associated virus is a small single-stranded DNA virus that's not associated with known human disease. It has a very simple structure, this small genome with these two packaging signals on the end. So this has been engineered to recombinant AAV, in which these packaging features of the wild-type virus have been replaced with a recombinant cargo, and then you just attach the packaging signals on the end. So this has no viral coding sequences, and the only viral sequences are the packaging signals. So this recombinant AAV is non-integrating. It produces a very small inflammatory reaction, which is transient, and the DNA is maintained as an episome for years. The main limiting feature is that the cargo can only be 4.7 kb. So there's several components to making the AAV that you would like to do your gene therapy. There's the promoter, the gene, the capsid. These all determine how your gene therapy product will behave. So there's a cardiac, some of these capsids are trophic to the heart, particularly AAV9, which you can see if you inject this systemically, you have a very strong signal in the heart. So AAV is coming, it's actually in clinical use now, and its use will only expand over the next several years. These are the clinical trials as of 2019 that have been done in gene therapy. You can see that some areas have had a lot more attention with gene therapy than others. The heart has received some attention. There's been one clinical trial for adult heart failure, which was not successful, largely due to dosing issues. There's actually 2 AAV gene therapy products on the market now in the US. One is Luxturna for eye disease, and the other is Zolgensma, which is really miraculous treatment for SMA. That's also the most expensive drug on the market. It costs $2.2 million per treatment, but it's a one-time treatment. So we looked into AAV gene therapy for Barth syndrome. So we characterized a novel knockout model of Barth syndrome in which the mouse gene has been removed. And this causes neonatal lethality in the mice. Interesting, you can stratify the lethality based on the birth weight of the mice, so that the smaller mice at birth have almost all of the lethality. This is just a picture of what the mice look like. They're smaller throughout their lives, and their heart function declines over the first several months of life. So we delivered AAV TAZ to these mice at birth. You can see we're getting efficient expression of the TAZ gene in heart muscle cells. And we can nearly completely rescue the lethality of these mice with the gene therapy. And we can also protect the heart from progressive contractile dysfunction. So we're very hopeful that the TAZ gene therapy could become a viable treatment for the patients. The main limitation that we've discovered so far is that the dosing is very important. So in our trials in the mice, you have to transduce over 70% of the heart muscle cells in order to get long-standing efficacy. So we can do that easily in mice, but in humans, that would still be a challenge at this stage. So just to summarize, we started with our patients and made our engineered tissue model. We discovered mechanisms that are important for the pathogenesis of the disease, and that's led to some new potential therapies. I'm going to shift gears now and go to the engineered tissue model of an inherited arrhythmia. Again, a close collaboration with the Parker lab, as well as my clinical colleague, Dominic Abrams, and Vasilios Bezaridis, who is both a clinical electrophysiologist as well as a cellular electrophysiologist. So again, we'll start with a case. So this was a 10-year-old girl who was in a swim race and lost consciousness. She was dragged out of the pool, and luckily there was a defibrillator there. She had 10 shocks, was resuscitated, and was brought to Boston Children's. She had a resting EKG, which was completely normal. But her exercise test was diagnostic. So here, with progressive exercise, she developed ventricular ectopy. Then this pathognomonic rhythm, which is bidirectional ventricular tachycardia. Then polymorphic ventricular tachycardia and ventricular fibrillation. Just so you know, we spliced this together to make it fit onto one thing, so it's a little bit synthesized. So this is a disease known as catecholaminergic polymorphic ventricular tachycardia. This is a ventricular arrhythmia that occurs during exercise or emotional stress. Typically presents in childhood with the average age of presentation about 10. At Children's, it accounted for 20% of unheralded cardiac arrest, and the incidence is thought to be 1 in 10,000. The mainstays of therapy are exercise restriction and beta blockade. Flecainide has found some utility in this disease. There's a surgical treatment in which you denervate the sympathetic innervation to the heart. And you can also use ICDs, although we're steering away from this therapy now, because if you get shocked when you're awake, you will induce catechol release, which will precipitate additional shocks, and this can be lethal. It's called electrical storm. Over 60% of these patients have mutation in the gene RYR2. This is the ryanodine receptor, which is the intracellular calcium release channel. In this one gene, there's over 160 different mutations that have been described to cause CPVT. I'm going to have to drag you through a little cardiac physiology now. So you'll recall that the action potential activates the L-type calcium channel on the membrane, which lets a little bit of calcium in from the outside, which then activates RYR2, which is on the sarcoplasmic reticulum. This lets the calcium in there come out into the cytoplasm and go to the muscle so that the muscle will contract. Then the calcium is returned to the sarcoplasmic reticulum by this calcium ATPase, and a small amount of calcium will go back out through the sodium-calcium exchanger. So the defect in this disease is that the RYR2 channel is somewhat leaky, particularly in diastole. So the diastolic calcium goes too high, which can allow the sodium-calcium exchanger to run, and that's electrogenic, so it can cause what's called triggered activity. So you get a beat, and then you get a little triggered beat afterwards. If this is big enough, it'll trigger a full beat or a cascade of beats. So again, we started with our patient. We made iPS cells, confirmed the mutation. We also took wild-type cells and put this mutation into the cells using genome editing. So then we took the individual cells and measured the calcium release, which in this picture is shown by these red dots. So the more red, the more spontaneous calcium release. And you can see that CPVT cells have a lot more spontaneous calcium release. And you can show this quantitatively, but this was not new. A lot of people had already done this. Our interest was making an engineered tissue model of the disease. So here we've taken a protein from algae that's sensitive to blue light, and we put it into the cells so that when we shine blue light on one spot, that those cells will depolarize. These cells are plated again in a way that makes them organized in a linear fashion. And at the end of these muscle strips, there's these little cantilevers that again will move when the cells contract. So here's what the assay looks like. You get a dish with all these little cantilevers moving in response to the light. And if we put a dye in that's sensitive to calcium, we can look at the calcium as it moves up the muscle strips. So remember, this is an exercise-induced arrhythmia. So our goal was to do an exercise test in a dish. So here we take the tissue, it's the same tissue, but we add more adrenergic stimulation, which is isoproterenol, and we change the pacing rate. And under conditions of simulated exercise, you'll see that the wild-type tissue conducts just fine. But we were very excited to see that the CPVT tissue at the highest levels of simulated exercise developed this abnormal calcium wave, which is known as a rotor. This is the tissue level equivalent of ventricular tachycardia. So here's a summary of almost a year's work on these tissue chips, showing that this is an exercise-induced phenomenon that's only seen in the CPVT tissue. We looked at these tissues before they had reentry to try to understand what made them vulnerable to this reentry. So we basically looked at every possible calcium conduction parameter we could think of. And in the gray area are the ones that were the largest change and the most statistically significant. So let's see what those were. It's the spatial dispersion of calcium speed, the temporal dispersion of calcium speed, and the diastolic calcium level. So essentially there's two things: the heterogeneity of the tissue in terms of its calcium waves, and the diastolic level of calcium. Those are the features that make the tissue vulnerable to arrhythmia. Now, if you're thinking like an electrophysiologist, you also think, in addition to the vulnerable substrate, what's the initiating event? So we can look at the initiating event by looking in detail at these traces of the CPVT tissue. So here's the same tissue. This is a tissue we paste at 2 Hertz with isoproterenol, and this is a velocity field. These are all little arrows, and you can see that they're nice and homogeneous. But if we just take the same tissue and go to 3 Hertz, you can see that we get a lot of heterogeneity in the velocity field. And then on this beat, we can see reentry happening with the vectors going in the wrong direction and eventually going backwards. So the interesting thing about this is that unlike the clinical impression of this disease as a triggered activity, here we did not see any triggered activity. This reentry was caused by a conduction block. So actually this was predicted by computational modeling, which showed that not only do these DADs cause triggered activity, but they can also cause heterogeneity of repolarization. So in these computational simulations, they showed that if you time a beat just right after a DAD, it will cause conduction block. So our data provides some experimental support to show that in CPVT, this is actually a relevant phenomenon. So we have a model of CPVT, but now we can ask, I think, the key question. Why is it that beta adrenergic stimulation precipitates the arrhythmia? What's the molecular link between exercise and arrhythmia? So we'll go back to this model, and the beta receptor is upstream of many kinases. 2 of them are CAM kinase and PKA. So each of these kinases has a lot of targets, and many of them have to do with calcium handling. So we hypothesized that maybe one of these kinases is the key player that links the beta receptor to the exercise-induced arrhythmia. So we took our cells and treated them with either PKA inhibitor or CAM kinase inhibitor, which I labeled here as P or C. And there's a lot of data, so I'll just jump to the conclusion, which is that the PKA inhibitor did nothing, but the CAM kinase inhibitor strongly inhibited the spontaneous calcium release. We also did this in our tissue chip model, and again showed that we can reduce the incidence of arrhythmia in 2 different CPVT genotypes by inhibiting CAM kinase. So then we can ask, what is the molecular target of CAM kinase? Is it one target or is it many targets? So we can ask this question using genome editing to see if we ablate a single target, could we protect the cells from the arrhythmia? So we made an educated guess. The channel RYR2 itself, which is the subject of the mutation, is well known to be phosphorylated by CAM kinase on this residue 2814. And this event is known to be arrhythmogenic. So we hypothesized that that phosphorylation event is essential for unmasking the arrhythmic potential of the mutation. So to test that hypothesis, we ablated this CAM kinase site in either the wild type background or in the CPVT background. We did the same with the 2808 just to be complete. 2808 is the target of PKA. Again, there's a lot of data. I'll just jump to the conclusion that ablating the PKA site had no effect consistent with the pharmacology. But ablating the CAM kinase site made the cells have a normal calcium release behavior. And then going back to the engineered tissue model, just changing this single residue can prevent these tissues from undergoing reentry. And this was true in all of the tissues that we tested. So I think this is, I mean, philosophically, I think it's both beautiful and tragic. You get one mutation that can make these patients at risk for a life-threatening arrhythmia, but a second single amino acid change can completely protect at least the tissue from the arrhythmia. So again, we went back and looked at the properties of the tissue that made them vulnerable to arrhythmia. And you can see that ablating that single phosphorylation site completely normalizes all of these properties of calcium conduction in the tissue. So our conclusion is that this phosphorylation event of 2814 is a key trigger of the arrhythmia, and that if we could prevent that phosphorylation event, perhaps we could protect the patients from arrhythmia. So that led us to think about CAM kinase inhibition and as a therapy for CPVT. This was done by Vasilios Bezaridis. So what do we know about CAM kinase in the heart? We know that actually it's important for fight or flight responses, just like the beta receptor. But just like beta blockers, well tolerated, you can actually ablate CAM kinase in the heart, and the heart, there's no adverse effects. Actually, it's protective against many forms of heart disease. However, CAM kinase is expressed in many other tissues, such as the brain, and you need it in your brain to make memory. So this has been the barrier to CAM kinase inhibition as a clinical drug. So we needed to inhibit CAM kinase selectively in the heart, which is where gene therapy comes in, because we can direct the expression of the inhibitory peptide only to the heart. So here we have our inhibitory peptide, and we use a cardiac-specific promoter to drive its expression in the heart. And here's some staining of different tissues showing the green color is where this vector is expressing. And this shows that we have targeted only the heart and not the other tissues, especially the brain. So we gave the gene therapy to a mouse model of CPVT. And then we characterize the calcium handling of the cells and the arrhythmia properties of the mice. So here's the cells. If you take normal cells and you pace them and then you suddenly stop the pacing, the mature cells will not have calcium waves for over 30 seconds. But CPVT myocytes have bizarre calcium waves at cessation of pacing. So we call these post-pacing events. And if you quantify this, you can see that CPVT myocytes have a high frequency of post-pacing events, which is normalized by our gene therapy. Then if we look at the whole animal, so if you just treat with catechols, you can induce ventricular tachycardia in the control-treated animals. But in our treated animals, they were resistant to arrhythmia. Vasilios also sees patients and does EP studies. So he did EP studies on these mice with a tiny octopolar pacing catheter that he inserted into the right ventricle. So here with ventricular stimulation, you can induce ventricular tachycardia in these mice, but the gene therapy completely protects them from the arrhythmia. So it seems that this is a promising therapy, but one important characteristic of a therapy that would be used clinically is that it should be independent of the genotype, because there's so many different genotypes that if it was genotype specific, you would not be able to bring that to market. So here's a picture of all the different mutations that have been described in RYR2. There's over 160 of them, as I said. They're the little black dots here. But they do cluster into 4 regions, which are the 4 different colors shown there. So we can't test all of the mutations, but we have tested a mutation representing each of these hotspot regions. And in each case, CAM kinase inhibition was effective. So to summarize, we started with our patient. We developed our engineered tissue model of the disease. We identified a molecular pathway that links exercise to the triggering of the arrhythmia. We then used that to develop a molecularly targeted therapy for the disease. And currently we're in the process of trying to translate this into a clinical therapy by partnering with several biotech companies. So I just want to take the last slide to reflect back on the use of iPS cells in personalized medicine. on the one hand, and in discovering new therapies for myocardial function. So I think you could think of it that we've rounded the first turn in that we now can robustly generate iPS-derived cardiomyocytes. We're pretty good at genome editing. We have proof of concept that disease models can yield insights that are useful in animal models at least. And we have some primitive physiological assays of heart function in the engineered heart tissue. But there's a lot of things that still need to be overcome. So there are many different types of cardiomyocytes, and we're not yet great at directing the iPS cells to form specific subtypes of myocytes. As I mentioned, the maturity of myocytes is a big problem. Developing methods for high throughput screening in these formats is very important, and that's something where Mark Marcola here at Stanford has really been leading the way. We need to determine if these cells really capture inter-individual variation or not, because a lot of the dreams of using them for personalized medicine hinge on that principle. And we need to develop better heart tissue and better physiological assays to characterize the tissue. With that, I'd just like to thank all the people that have worked on this, particularly my close collaborator, equal partner in this, project, which is Kip Parker. Thank you.
That was absolutely amazing. Given the fact that plasmid mutations cause such disruption of sarcomere structure, do you think, and do you have any data perhaps suggest that once a patient clinically presents, which is not immediately at birth, although sometimes those children are hypotonic, if they present at, say, a couple of months of age, any data that you can reverse some of the damage once it's occurred?
Yes, right. Dan is asking whether gene therapy would be effective in Barth patients after they present with heart disease because, for example, the sarcomeres may be permanently disordered. So we have looked at that. Actually, in the mouse model, by both light microscopy and EM, we don't see a sarcomere defect, which might mean that there's a difficulty in assembling sarcomeres that we can see in the iPS cells, but not in in real mice, which is the same as has been described for Titin mutations in DCM. But we have looked at that actually in our gene therapy program for the Barth mice. And we can reverse established mild heart dysfunction, but whether we can reverse severe heart dysfunction is not, we didn't look at that. The main concern is that we actually see an increased apoptosis rate in the Barth cardiomyocytes. And obviously, if you have death of too many myocytes, you can't come back from that. Other questions?
Or you mentioned and pictures of the sarcomere look pretty good. At first glance, it's not how you solved it, but still an issue. And I'm wondering what the current challenges are. So, how repeatable are you? Now you're talking to pharmaceutical companies that or recruitment.
Right, so the maturity is a major issue still. Sarcomere, the appearance of the sarcomere by those light microscopy images is a crude metric. So there's many other metrics, including the mitochondrial organization, the metabolism, the channels that are expressed by the myocytes. So in all those cases, they do not perfectly replicate adult human myocytes. But they're, we're getting closer, and everyone is trying different, including many of the people here at Stanford, are trying different maneuvers to improve the maturation of the cells. In terms of whether they're being used yet by pharma, yes, they are. All the pharma companies are using this now as a drug development platform. I think more in a high throughput format. We sort of envision not only the high throughput uses, but also once you have a candidate therapy, you could think about de-risking a clinical trial by making more elaborate engineered tissue models that might test functions that you would not detect in a single cell assay. Yes?
One of the issues with using viral vectors in gene therapy right now is whether it's in the liver issue. Can you comment a little bit on AAV9?
Yeah, that's a great question. So I think the primary toxicity that's been seen so far in large animals, not really in mice, is an acute inflammatory response that involves the liver and that can, at a high enough dose in primates has precipitated lethal thromboembolic events. That's thought to be an acute reaction to the capsid, and it's probably what will limit the dosing besides manufacturing and just feasibility. That is probably the dose limiting aspect right now. And that's important because for muscle diseases such as muscular dystrophy or BARF, transducing a high fraction of cells is very important. So we're probably, there's the competing interests of getting enough cells versus the acute toxicities that you mentioned. In the heart muscle itself, we don't see much evidence of inflammation. Okay, thanks.